Hello students, welcome to Biotech Academy. So we've discussed about we are discussing about Mendelian basis of inheritance theory which Mendel proposed, and we've discussed about monohybrid cross, and today we'll talk about dihybrid cross. So what is dihybrid? So di means two and hybrid. So when Mendel was studying the inheritance pattern of 7 pairs of contrasting features in Pisum sativum, so first he looked into one character at a time, how that character is passing from one generation to the other. He looked into the inheritance pattern of one character at a time. Now so that was monohybrid cross. Then he looked into two contrasting features at a time. So that is dihybrid cross. And according to the dihybrid, based on this dihybrid cross, he proposed a law which is known as a law of independent assortment. So according to this law, law of independent assortment. Alright, so according to this law, factors, or when Mendel was postulating his theories, at that time he did not, the term gene was not coined, and he proposed the genes to be factors. So he said that there are certain factors which are responsible for a specific phenotype or a specific characteristics appearance. So then when he looked into two characters at a time, so according to the law of independent assortment, it states that factors for different pairs of contrasting features means factors which are responsible for different features. So one factor is associated with one specific phenotype or one specific character. So according to the law of independent assortment, it says that different factors which are responsible for different contrasting phenotypes or contrasting characters, they are distributed independently into the gametes. So during the gamete formation, the factors, because they are in pairs, so they are in 2n configuration. So when they form n half during meiosis, so then he mentioned that those formation, the separation or the segregation pattern of each pair is independent of the other. Alright, so then to explain this law, he did the dihybrid cross, and he took into account two features at a time. One was seed color and seed shape. So he took into consideration, in dihybrid cross, he took two features at a time, seed color and seed shape. Okay, so seed shape, there are the two contrasting features are round and wrinkled. Either the seed will be round or it will be wrinkled. So round is dominant and wrinkled is recessive, and we've already discussed about what is dominant and what is recessive. So in seed color we have two types, yellow and green. So yellow is dominant and green is recessive. Alright, so let's see how the dihybrid cross Mendel did and explained. So he took two features and he did the crossing between two pea plants which had different opposite. So if one is round, then the other would be wrinkled. The seed of the other plant would be wrinkled. So he crossed this way. So he selected two pea plants. So that is parental generation, P generation. In P generation, he crossed a completely dominant plant with like both the features were dominant, to a completely recessive one. So he crossed, so this was the genotype of the parental generation. Okay, so it's round and yellow, it's wrinkled and green. Okay, so he made a cross. So how the gametes will be formed? So if this is segregating this, so these factors, they always appear in pairs, and the pairs they separate during gamete formation, and then during hybridization then they restore the pair form. Alright, so during gamete formation, so the segregation pattern of this round of the factor which is controlling the seed color, seed shape, and the factors which is controlling seed color, the segregation will be independent of each other. So because it's a dihybrid cross, so one of this and the other of this. So that's how the gametes will be formed. So I'll elaborate over here. Okay, so this is the one parental generation, genotype of one parental generation. So how the gametes will be formed? This R and this Y. Again, if you keep this as constant, then it will vary with this R, this Y. If you keep this one as constant, then again you will get this R, this Y. And if you cross this one with this one, like if they segregate from, and during the gamete formation this R comes with this Y, then we have this. So every time, because the genotype is such that both the factors are same, both the pairs are the same, so every time you are getting this one. So for ease, just to make clarity of this, so we get this is the gamete. Similarly, the gamete formation will be over here. So this R with this Y, this R with this Y, this small r with this small y, this small r with this y. So every time we will get this as the gamete. So this is in the case when they are doubly recessive or doubly dominant. Okay, so when they hybridize or they fuse, so we get first filial generation, or we call it as F1 generation. And in F1 generation we get, okay, so now because this seed in this one, because this seed shape which is round is dominant over wrinkled one, so the phenotype of this will be round. And for similarly for seed color, so yellow is dominant over red, yellow is dominant over green, so we'll get yellow. So this is the phenotype of the F1 generation. But then because it's an F1 generation, in F1 generation you did not see any recessive phenotype here, so that's why he went on to F2 generation. This logic was there again in the monohybrid cross. So similarly now, then what he allowed selfing of this one, and then gamete formation, then he looked into the gamete formation and then the phenotype. Okay, so for this we'll just see how gametes are formed for this one. So we have this as our F1 generation. Then he allowed selfing. This is F1 generation. So this is represented, remember which square? This is Punnett square. So in Punnett square, now how the gametes will be formed for this one, because both this R with this Y, this small r with this Y. So let's see how the gametes are formed. We have both, because it's selfing, so both, one, suppose if this is male and this is female, so we'll write the gametes in the respective columns. So the gametes will be, if this R with this capital R with capital Y, capital R with small y, small r with capital Y, and small r with small y. This is for one sex. For the other sex, similarly, the gametes will be like this: capital R with capital Y, capital R with small y, then small r with capital Y, and small r with small y. Now, fusion. So the genotypes are like this: capital R, small r, capital Y, capital Y. So this is how okay, now let's see, these are the various genotypes of the 16 plantlets that were obtained, and then we'll just see how the phenotypes of each one. So because always remember, this capital is for dominant. So if this is like this, then we get round and yellow. Again round, yellow, round, yellow, round, yellow, round but green, round, yellow, round, green, round, yellow, round, yellow, round, yellow, wrinkled, yellow, wrinkled, yellow, round, yellow, round, green, wrinkled, yellow, wrinkled and green. So if we count, then the phenotypic ratio becomes 9:3:3:1. So this 9 is for the, and this 1 are for the parental phenotypes which were taken initially. So this is for the dominant one. So round, 9 plants were with round seeds and yellow colored seeds. One plant which was obtained was with wrinkled seed which were green in color, and these two, 3 and 3, they are the hybrid phenotypes which were obtained. So round seeds, but then the seed color was green, or wrinkled seed shape with green, with yellow phenotype, yellow color, seed color. So this is about dihybrid cross. So you need to remember, then he checked the inheritance pattern of other pairs, or other 7 pairs which he selected for his studies, and every time the phenotypic ratio was 9:3:3:1. So this is the phenotypic ratio, out of which this and this one, because these are the numbers where the parental phenotype was obtained. So this is round and yellow. This is wrinkled and green, and this one is for cross. So round seeds, although the seeds are round, but then the color is green. This is for, although the seed shape is wrinkled, but the seed color is yellow. So this is how he studied the dihybrid cross, and then we'll just in the next lecture we'll discuss about chromosomal theory of inheritance, and which follows the similar pattern like the dihybrid cross over here, which we have discussed just now. So you can go through the reference and you can come up with your any doubts or queries. You can mail me or you can ask in the comment section, and I'll be more than happy to answer all your queries and doubts. So please hit like and share among your friends, and do subscribe for similar videos.